Thank you so much again, Helen, for coming on with us. We really appreciate your time today and letting us be part of this fundraiser. Yeah, could you let us know a little bit about your background and what you do now at the Hutch before we get into some of the science?
Oh, yeah. So I started working at Fred Hutch as a scientific content liaison. So essentially that means that I work with all of the researchers, the scientists, the clinicians at Fred Hutch, and I try to understand what their work is. So I try to figure out what are they doing, what are they researching, what clinical trials are they running, what patients are they meeting with. And then I communicate that understanding to the community. So sometimes that's by speaking myself, sometimes that's by arranging relationships between our scientists and clinicians and our community partners. And I also work with our philanthropy team to help create educational content and to put on fundraisers so that we can finance all of our beautiful, amazing researchers. Because we are a nonprofit, which means we got to find money somewhere. And a lot of that comes from our Fred Hutch community.
And what,
Yeah,
so how difficult is that in the current funding climate? Just to like illustrate to the chat why something even like raising money on Twitch is a meaningful difference to the researchers that you're coordinating with.
It is easier to get funding for some things than others, right? And again, I'm brand new, so I'm sure that someone with more experience would be able to speak more eloquently to this. But we all want to fund cancer research. We all know someone who has been impacted by cancer. Many of us ourselves have had cancer, and we all want that to be less of a burden on individuals and on society. Same with infectious disease, which is another one of the specialties at Fred Hutch. You know, we all want that to be less of a concern for humanity. But there are so many different causes that are demanding our attention, that are clamoring for need, you know, environmental causes, political causes, social justice movements. I mean, there's so much that we need to care about. I think it is hard to find that. And as a nonprofit, and especially as a nonprofit that encourages new scientists who might not get funding otherwise, scientists who are pursuing really bold ideas, really innovative ideas, they're less likely to get funding from more traditional routes. And so we need these grassroots efforts where we can find funding to fund innovation from folks who might not otherwise find that kind of support.
And that's because it's a risk, right? Like if I'm innovating, like I want to work on spiders to figure out, I don't know, how physics work or something, but I've only ever worked in fruit flies. That's a big investment and a big jump, and that might not pay off. So you're not going to maybe get a federal grant. But if you have an organization that's a nonprofit that's like, okay, you actually have a solid set of ideas, let's give it a shot, that's where that funding is going to be coming from.
Right. And also like younger scientists, right? So like you and Lita were both, you know, postdocs. And so you understand that it can also be hard for like folks that don't have a lot of clout behind their name yet to find funding. You know, that can be tricky. So one of the things that we talk about often in a genetic counseling setting is that all cancer is genetic, but not all cancer is hereditary. So first we're going to address that genetic component, and then we'll talk about sort of that hereditary component. Hereditary just meaning inherited. So something that runs in families. So we know that cancer cells have different biology than our typical cells, than the cells that populate most of our body. And we know that they also have different genes, that the genetics of a cancer cell are very specific to cancer cells. And there are two specific classes of genes that we were saying are important. We talked about genes as being chapters in the genetic instruction book that is our DNA. And each of those chapters has a specific job. Each of those genes has a specific job in our body. And we were talking about the first category of genes, which is our tumor suppressor genes, and how these genes have jobs that help our bodies to suppress tumors. When these genes are working properly, they help to prevent tumor growth. They help to prevent cancer. A lot of times that's because these genes work as DNA repair mechanisms. And so if our DNA gets damaged as we go through life, it acquires these, mutations that could be harmful. When our tumor suppressor genes are working, they go, not on my watch, baby, and they fix that mistake before it can cause the cell to become cancerous. So we think of these as the brakes of a car, because if the car goes way too fast, it's going to hit a wall, and that wall is called cancer. And we want our car to be driving along at a healthy rate that causes it to be a healthy cell that is growing and dividing, but not in a cancerous way. So we think of these as the brakes because they help to stop that car when it starts to go too fast. And you've seen that I made a little note there that loss of function variation is most likely to cause these genes to, be involved in cancer. And that's because these genes you want to work. And you usually have 2 copies of each, again, one from our mom or the egg and one from our dad, the sperm. And we want them to work, right? These are tumor suppressors. We want our body to suppress tumors. So anything that damages these genes in a way that causes them to lose their function, called loss of function variants or loss of function mutations, when those get lost, that causes damage. Now, that's our first class. All right, so tumor suppressors, again, we usually are born with 2 copies and we want them to work. We love them. Proto-oncogenes are a little different. Proto-oncogenes, these are genes that usually they have a job, usually involved in cell growth and division, and they help our cells chug along. So we think of them as like gas for the car. You gotta have it, but you only wanna have a little bit. These genes, if they have what's called a gain of function mutation, so different from our tumor suppressor genes, which usually have loss of function mutations when they're involved in cancer progression, if they have a gain of function mutation that causes them to be overly active, it's like having, I don't know enough about cars. I don't think you can have too much gas. I don't think that's a thing, but it's like having a brick on the gas pedal. And it causes the car to, again, accelerate into that wall that we're calling cancer. It causes the cell to grow too quickly, to become cancerous, to evade the immune system. It promotes all of these different changes in that cell's biology, which cause it to become pathogenic. And so either one of these kinds of variants or mutations is common in cancer progression, and they're both really important targets right now in the way that we understand cancer progression and the way that we understand cancer cause, and the way that we hopefully try to target and treat cancer and identify folks who are at an increased risk for cancer, and therefore we can start screening them earlier and do specific kinds of treatments that are recommended for folks that have a predisposition for cancer. I feel like for many people, understanding the biology of cancer makes the experience less scary because it feels less like this horrible chance event that happened, and it feels more like this horrible biological phenomena that has happened that we are understanding that we can try to manage, right? And I think that when people feel like there's less mystery about it, it's also less scary. Maybe not for everyone, but I think for many people. So I think that it's nice to have these resources for those who wish to know more information, and those who don't, obviously just like focus on getting through it and block that out, like live your life and you don't have to know this. But for folks that do find it helpful, I think it'd be nice to know.
Yeah, we've talked a little bit about that with the Undiagnosed Disease Network, where it's like one kid, maybe, or 3 kids will have it a year, this like certain mutation. That's not enough for anyone to research it because it's not going to make money from a pharmaceutical perspective. But then there's researchers who sequence the genome, they put in the mutant copy in a fruit fly and figure out what's wrong. And just even if there's no treatment, just knowing what's wrong can be very, very empowering. Yeah.
There are kind of 3 general categories of cancer that we often talk about. The vast majority of cancer, about 70% of cancer, is what we call sporadic. So this basically just means that as you've gotten older, your cells have had time to acquire mutations. And these mutations are either in tumor suppressors or proto-oncogenes or other genes associated with cancer. And that cell becomes cancerous. What does that mean? That means that that cell is now growing and dividing unchecked. It's unhinged. It is evading the immune system because your immune system, it wants to destroy precancerous cells. Your T cells and other cells in your immune system, they can sense a precancerous cell. They're like, that's not what I want. I don't want that. That's growing funky. That looks strange. And it will try and destroy it. But cancer cells, because of all these genetic changes, they're able to evade the immune system. They're able to grow where they're not supposed to grow. They're able to make themselves look enough like a normal cell, or enough like a move quick enough, move in a specific way, metabolize in a unique way, different from your normal healthy cells, that allows them to grow the way they do. So this is what that looks like in your body. So you're born with like all of your tumor suppressor genes, proto-oncogenes, genes involved in sort of the immune system. All of these cells are genes are working the way we'd expect. So that's far left, you're born. And then over time, let's say one of those cells loses a tumor suppressor. Don't worry, we got a backup. Ain't no thing, we got a backup, we're fine. That's why you have 2 copies of each, we're good. But then you go through life, more aging, more UV, more, bad luck, what have you. and both copies of the tumor suppressor are lost. Now that cell is not as good at preventing tumors because it's lost its tumor suppressors. And so that gene that cell over time accumulates more damage. It accumulates more mutations, more variants that cause it to become cancerous. And then you see that cancer spreading. So that's the vast majority of cancers. So there are two more categories of cancer. The next biggest category, about 10 to 20%, is what we call familial. And this most likely means that there are sort of multiple genes in a family, so genes that we share, specific versions of a gene, that put us in increased risk for cancer, as well as environmental factors that we might share as a family that put us in increased risk for cancer. So, for example, a family that grew up in a certain region that was exposed to a lot of chemicals that were later found to be associated with cancers. So these folks, we see in their family histories that there's a greater chance that these folks are going to have cancer. We're seeing more cancer than we'd expect in the family. But if we do genetic testing on these folks, we're not going to come back with a change in a gene that we know is associated with cancer, like BRCA1 or BRCA2, which are associated with hereditary breast and ovarian cancer syndrome. And that's because these genes are sort of lower risk factors, right? They increase your risk for cancer, but not as much as what we call the high risk cancer genes, high penetrance cancer genes, which are those hereditary cancer genes, which is our smallest subset. So we've got sporadic, which is most, familial, which is sort of in the middle. And then our smallest subset, about 5 to 10% of all cancers, what we call hereditary. So when we talk about like BRCA1 and BRCA2, for example, which are associated with hereditary breast and ovarian cancer syndrome, so a form of hereditary cancer, that smallest category, that 5 to 10% of all cancers, these folks, instead of being born with 2 copies of BRCA1, which is a tumor suppressor, instead of being born with 2 copies of that gene that work and help suppress tumors, they're only born with 1 copy that works. Typically, that's because one of their parents, one of their biological folks, either their mom or their dad, the egg or the sperm, didn't have a copy that worked. So usually hereditary cancer runs in families. We often see multiple generations affected. And that's because they're usually inherited or hereditary. So these folks, they're born with only 1 copy. And that means that they're at an increased risk to develop cancer because they only need to lose 1 more copy in order to get to that cancer stage. Most of us have to lose both copies before we get to that cancerous stage. But these folks, because they're born with 1 copy that doesn't work quite right, they only have to lose 1 more copy. And then they're at that cancer stage.
Is that something that you can test in an embryo? Like if can you genetically counsel 2 folks, like, you know, who are soon to be parents and be like, can you test on the embryo to make sure that both copies are normal before you proceed with implantation, let's say?
Is this a leading question? Do you know about PGT?
Possibly.
Yes. So let's say we'll just come up with a scenario. Let's say that I am looking to have a child with a biological partner, and I want to be the egg donor, and I want my partner to be the sperm donor. And we find out that I carry a genetic condition, and that I'm a carrier. So we use the term carrier to refer to someone who, as with hereditary cancer, has 1 copy of the gene that works and 1 copy of the gene that doesn't. Typically, carriers don't have any symptoms. They don't know that they're carriers. They have no idea that they carry this not working copy because they're totally fine. They've got this backup. For some conditions like hereditary cancer, you might know because hereditary cancer is often associated with cancer at younger ages, more cancers in a family than you'd expect, and specific kinds of cancers that are more likely to be hereditary than others. So there might be things in your life that might suggest it, but generally asymptomatic in terms of you've developed to adulthood without any major health concerns. So let's say I'm a carrier, and let's say that my partner and I don't want to take any chances that our child will be affected with this condition. It's a deeply personal decision. There's no right or wrong, but let's say in this scenario, that's the decision that me and my partner have made. We could do IVF, where they take the eggs out of me, they take the sperm out of my partner, and then in a petri dish, the 2 shall meet. And then we have a fertilized egg. We have our little embryo. And what they can do at around day 5 or 6 of development, they can take a little sample from that embryo. This is called pre-genetic testing, PGT. And they can actually look at those small subset of cells they've taken from the embryo. The embryo then goes and continues to grow, or they'll freeze it while they do the testing on the little sample. So they'll freeze that so that it gets held. And then they'll do that testing on the small sample, and they'll look to see, do we see that this embryo has that same pathogenic or disease-causing change that her mother carries? Or is this embryo, did it not have that? Because again, if you've got one copy that works, one copy that doesn't, 50-50 chance that it gets passed down to each child, to each egg, you know? So 50% of them might have the change, 50% of the embryos might not. And then we could selectively implant an embryo that was not found to have that change. There are caveats, of course, but that is the general idea.
And what's cool is too, like in studying the social insect side of things, you can see the effect of social setting on tumor progression, because there's like all these meta-analyses on humans where it's like, if you live alone, you're more likely to get a more aggressive form of cancer. If you're very crowded, maybe you get a more aggressive one too. But it's these meta-analyses, right? So it may or may not be biologically relevant, but you can test that hypothesis in an insect and then maybe extrapolate, like we talked about with Anderson, all the way from yeast to human cells, from here insects to humans as well.
Excellent.
So I know you have to go have dinner, but is there anything like rounding out that we'd like to get to, or a topic that you really wanted to still highlight? I think we cover like a really good set of like the inheritance pattern of cancer, which I think is a fundamental that hopefully people identify with and connected with, along with just like mutational rates and what that relates to from the perspective of a genetic counselor, which I thought that was incredible. Thank you for that explanation. The car analogy, really, really good. Really makes sense. Hopefully it did for y'all as well.
No, this was lovely. This was so fun. Thank you for letting me come and chat and stay so long. I love talking about science always. I love talking about genetics always. But no, this is just wonderful. I think that like what makes organizations like Fred Hutch special is community, and like seeing the community that y'all have here. Like this is what science should be, right? Like science should be people being safe, being inclusive, being welcome, being warm, being excited. Like that's what it should be. It should be fun and goofy and interesting and challenging. And I love that. You've created something really, really special. So thank you for letting me share it with you.
Thank you so much for that. I think that means the world to us. And Helen, thank you again for your time, and have a lovely dinner from chat.
All right. Bye, y'all. Thank you.
Bye, Helen. Thank you.